Hey everyone, it's been a long time since I have uploaded a video and I apologize for that. So let's get back to the new lesson. So in this lesson today we are going to learn about electron transport chain. So before I talk about electron transport chain, I would first like to give you an overview of cellular respiration. So cellular respiration can be broken down into 4 different stages. In the first stage, which is the glycolysis, so as the name suggests, glyco means glucose and lysis means breaking it down. So glucose is broken down into 2 molecules of pyruvate, and in the process it generates this 2 NADH. Now, the second stage is the oxidative decarboxylation of pyruvate to coenzyme A. So the pyruvate is oxidized into acetyl coenzyme A. In the process, it also generates NADH. And now the third stage is the oxidation of acetyl coenzyme A to carbon dioxide. So in this process, acetyl coenzyme A is oxidized into carbon dioxide through tricarboxylic acid cycle or Krebs cycle, and the electrons which are liberated through this process, those are passed on to the coenzymes, that is NAD and FAD, and these coenzymes are then reduced to NADH and FADH2. Now the final stage is the electron transport chain and oxidative phosphorylation, where NADH and FADH2 give up their electrons to electron transport chain, and through that process ATP is synthesized or produced. So these are the 4 stages of cellular respiration, and it is also important to know the location of each of these stages. So basically glycolysis takes place in the cytosol, and pyruvate is oxidized to acetyl coenzyme A in the mitochondrial matrix, where as well as the oxidation of acetyl coenzyme A to carbon dioxide also takes place in the mitochondrial matrix. And the last stage, which is the electron transport chain and oxidative phosphorylation, that takes place in the inner membrane of the mitochondria. Now, I just want to clarify one thing, that in my previous video I have mentioned that the cellular respiration is broken down into 3 different stages. That is just because I combined the first stage and the second stage. That's why I mentioned as 3 different stages, but I broken it down into one. I broken it down and divided that further into 4 stages. So I just wanted to clarify that with you so that you don't have any confusion. Now, when we learn about electron transport chain, it is important to know that how the electrons are carried through electron transport chain and oxidative phosphorylation. So there are 3 types of electron transfer takes place in oxidative phosphorylation. First, there is a direct transfer of electrons, as in the form of reduced ferric ion to ferrous ion. So when the electrons are passed on to ferric ion, it is then reduced to ferrous ion. And electrons can be also transferred at hydrogen atom, that is hydrogen atom, one hydrogen atom and 1 electron, and it can also be transferred as a hydride ion which bears 2 electrons. So this is something I thought it is important to know. And lastly, I also quickly want to talk about the structure of mitochondria, which is important before we start learning about the electron transport chain. So mitochondria has an outer membrane, and this outer membrane contains a special pores which making it freely permeable to most ions and small molecules. So this is the outer membrane, and there is this inner membrane which is a specialized structure that is impermeable to most small ions, including hydrogen, sodium, potassium, as well as most small molecules such as ATP, ADP, pyruvate, and other metabolites. And the inner membrane is unusually rich in proteins, and half of which is directly involved in electron transport chain and oxidative phosphorylation. So you can see that all the enzymes involved in electron transport chain and oxidative phosphorylation that are located or embedded into the inner membrane inner membrane of the mitochondria. And another important thing to know about inner membrane of the mitochondria, that it is extensively folded, and this extensive folding is called cristae, and this is important to increase the surface area of the membrane. Now next is the intermembrane space, which is just the space between the inner membrane and the outer membrane. And last is the mitochondrial matrix, which is the space inside the inner mitochondrial membrane, and it also contains most of the enzymes which are involved in TCA cycle, as well as the enzymes involved in fatty acid oxidation. So this is the structure of mitochondria, and now let's start talking about the electron transport chain. So the first enzyme in electron transport chain is NADH dehydrogenase, and it is also called as complex 1. Now, complex 1 or NADH dehydrogenase is L-shaped enzyme, and it is a transmembrane protein with one arm embedded in the inner membrane and the other arm extending into the mitochondrial matrix. Now it contains a molecule of coenzyme, which is flavin mononucleotide. So complex 1 is tightly bound to coenzyme FMN. And it also contains iron atom, which is also paired with sulfur atom to form the iron sulfur centers. And most importantly, it transfers electrons from NADH to coenzyme Q. And now we are going to learn about how the electron transfer takes place through complex 1 to coenzyme Q. So I would like to remind you again that acetyl coenzyme A is oxidized into carbon dioxide through TCA cycle, and during the process the electrons which are liberated, they are passed on to coenzymes, and these coenzymes are then reduced to NADH and FADH2. So the electrons are actually carried by NADH and FADH2. So you know that where the electrons are coming from. So in the first step, the free proton and the hydride ion are carried by NADH, are first transferred to complex 1 of NADH dehydrogenase enzyme, and the electrons from the NADH are first transferred to the tightly bound FMN coenzyme. And when the electrons are transferred to FMN, FMN is then reduced to FMNH2, and during the process NADH is also oxidized to NAD+. Now I also mentioned that complex 1 also contains iron sulfur centers. So in the second step, the electrons from FMNH2 are then transferred to series of iron sulfur centers. And now in the last step, which is the third step, the electrons from the iron sulfur centers are then transferred to coenzyme Q, and when the electrons are transferred to coenzyme Q is reduced to QH2, and as a result FMNH2 is then oxidized and converted back into FMN. So basically what complex 1 does is it actually transfers the electrons from NADH to FMNH2, and through iron sulfur center it is then transferred to coenzyme Q and reduce the coenzyme Q to QH2. So now the electrons are with coenzyme Q. Now I also want to mention some important things about coenzyme Q. And coenzyme Q, first thing, coenzyme Q is also known as ubiquinone, and keep in mind that coenzyme Q is a mobile electron carrier. Basically what it does is it actually moves to complex 1, picks up the electrons, and then transfer to the next member of the electron transport chain. And remember that electrons have a negative charge, and on their own they cannot move along the inner membrane. And the membrane is actually made up of phospholipid, and the tails of the phospholipid are nonpolar, and they don't interact well with negatively charged electrons. So basically the electron has to be carried by a mobile electron carrier. So in short, it is important to know that coenzyme Q is a mobile electron carrier which is embedded in the inner membrane, opposed to the complex 1, which is an integral protein located in the inner membrane. Now the other important thing to learn about complex 1 is when the electrons are passing through complex 1, it becomes supercharged, and the enzyme doesn't like to be supercharged. So what it is going to do is it is going to pass its electrons to coenzyme Q. So when the electrons move through complex 1, they liberate some energy, and which causes the conformational change in complex 1. As a result, complex 1 acts as a proton pump and transports 4 proton across the inner mitochondrial membrane. That is, the protons are transferred from the mitochondrial matrix into the intermembrane space of the mitochondria. So this is something important to know, that when the electrons are passing through complex 1, it is liberating energy which causes the conformational change in complex 1, which makes the complex 1 acts as a proton pump. As a result, complex 1 is going to transport electrons from mitochondrial matrix to intermembrane space. So some important thing about complex 1 is it oxidizes NADH into NAD plus and reduces coenzyme Q to QH2. Therefore, complex 1 also called as NADH ubiquinone oxidoreductase. And another important thing is it is able to pump protons across the inner membrane, that is from mitochondrial matrix to intermembrane space of mitochondria. Now let's talk about complex 2, which is succinate dehydrogenase. As it is titled, it is known as succinate dehydrogenase, and it is the only TCA cycle enzyme in the inner mitochondrial membrane. So let me remind you that when acetyl coenzyme A is oxidized to carbon dioxide, one of the steps of TCA cycle is to convert succinate into fumarate, and during this process FAD is reduced to FADH2. So the electrons from FADH2 are then transferred to complex 2, and then we will see how the electrons are then carried through complex 2 to coenzyme Q. So basically this is the only TCA cycle enzyme which is located in the inner mitochondrial membrane, as opposed to what we just learned, that majority of the TCA cycle enzymes are located in the mitochondrial matrix. Now it also contains iron sulfur centers or iron sulfur protein, and importantly it transfers electrons from FADH2 to coenzyme Q. Therefore it is also known as succinate coenzyme Q reductase complex, because it reduces coenzyme Q. So now let's learn how the electrons are transferred through complex 2. So basically we just learned that in TCA cycle, succinate dehydrogenase or complex 2 enzyme convert succinate to fumarate, and during the process FAD is reduced to FADH2. So now the electrons are with FADH2, and the electrons from FADH2 are then transferred to again series of iron sulfur centers, and electrons from iron sulfur centers are then subsequently transferred to coenzyme Q, which then reduces coenzyme Q to QH2, and as a result FADH2 is then oxidized and converted back to FAD. So basically here we have electrons from the reaction where succinate is converted into fumarate, and it is then transferred onto FAD, and from FAD it is then transferred to iron sulfur center and eventually to coenzyme Q, which reduces coenzyme Q to QH2. And this is the electrons from NADH, which is also transferred to coenzyme Q. So basically both complex 1 and complex 2 transfer their electrons to coenzyme Q, or in other words coenzyme Q accepts electrons from both complex 1 and complex 2, which is important to know. Now the another important difference between complex 1 and complex 2 is when the electrons are passing through complex 1, the energy which is liberated by passing through electrons is very small, and which is not sufficient enough to cause the conformational change in complex 2 to act as a proton pump. Hence it does not pump proton across the inner membrane and does not contribute to transport of transfer of protons across the inner mitochondrial membrane. Again, the complex 1 does not pump protons, and complex 2, because it reduces coenzyme Q, it is also known as succinate Q reductase complex. So now let's talk about complex 3, which is also known as cytochrome BC1 complex, and is actually a dimer, and each monomer consists of 3 proteins. So first is cytochrome B, which contains 2 heme groups, and second is cytochrome C1, which contains 1 heme group, and lastly is the risky protein, which contains 2 iron and 2 sulfur groups. Now, the important thing about cytochrome BC1 complex is it transfers electrons from reduced coenzyme Q, that is reduced QH2, to cytochrome C via complex 3. And this electron transfer is achieved via a unique redox pathway which is called the Q cycle. And I will have a separate video on Q cycle if anybody is interested to learn that mechanism in detail. And complex 3, also known as ubiquinone cytochrome C oxidase, because it oxidizes coenzyme Q or ubiquinone and reduces cytochrome C. So therefore it is called as ubiquinone cytochrome C oxidoreductase. So at this stage, the electrons are with coenzyme Q in the form of reduced QH2. So the electrons from QH2 are first passed on to the Fe+3 of complex 3, which is the ferric ion, and once the electrons are passed on to ferric ion, it is then reduced to ferrous ion, that is in Fe+2. As a result, reduced QH2 is then oxidized and converted back into coenzyme Q. One important thing to remember that from here all the complexes are going to be cytochrome complexes, and they contain this iron atom in the center of the porphyrin ring. So this is the porphyrin ring which contains these pyrols, and this iron atom in the center of the porphyrin ring that shuttles between the ferric ion to the ferrous ion state, which is common in all the cytochrome complexes we are going to talk about from here. And also remember that anytime you have a loss of electrons, oxidation occurs, and if there is a gain of electrons, reduction occurs. So in this case, QH2 loses its electron, so oxidation occurs because it is oxidized to coenzyme Q, whereas Fe+3 or ferric ion, it gains electrons, so it actually reduced to Fe+2 or ferrous ion. So anytime there is a loss of electron, oxidation occurs, and if there is a gain of electrons, reduction occurs. That is something important to remember. Now from here, the electrons which are now with ferrous ion, that is Fe+2, are then transferred onto cytochrome C or are then transferred onto Fe+3 of cytochrome C, which is then reduce the Fe+3 into Fe+2 of ferrous ion. As a result, the ferrous ion on complex 3 is then oxidized and converted back into Fe+3. And most importantly, when the electrons are transferred through complex 3, it becomes supercharged, and therefore it causes the conformational change in complex 3 to act as a proton pump, and therefore complex 3 also transport 4 proton across the inner membrane, that is from mitochondrial matrix to intermembrane space. So basically complex 3 pumps protons across the inner membrane, and because it oxidizes ubiquinone and reduces cytochrome C, it is also known as ubiquinone cytochrome C oxidoreductase. Now one important thing about cytochrome C is, just like coenzyme Q, it is also a mobile electron carrier, but it's a surface protein as opposed to coenzyme Q, which is an integral protein. And also remember that it is loosely associated to the cytosolic side of the inner membrane towards the intermembrane space. It is loosely attached, that's why it is actually a mobile protein, so that it can move from one complex to other and carries electrons. Now next is the complex 4, which is called as cytochrome C oxidase. It carries electrons from cytochrome C to molecular oxygen. It also contains copper ions, that is copper A, copper B, and 2 heme groups, heme A and heme A3. Now at this stage, the electrons are with reduced Fe+2 of cytochrome C, and from here they are then transferred to Fe+3 of complex 4, which then reduces Fe+3 into Fe+2, and as a result, reduced Fe+2 is oxidized and converted back into Fe+3. Now the electrons from Fe+2 of complex 4 is then finally transferred to the oxygen atom in the mitochondrial matrix, and once the electrons are transferred to oxygen, oxygen is then reduced to form water in the mitochondrial matrix. as well as it also oxidizes Fe plus 2 and convert it back to Fe plus 3. Now when the electrons are transferred to complex 4, it is again supercharged and causes the conformational change in complex 4, making it act as a proton pump to transport 4 protons across the inner membrane space. That means from the mitochondrial matrix to the intermembrane space. And this is also the reaction of reduction of oxygen, which forms the water. So you have oxygen, then 2 protons, 2 electrons, which then forms the water in the mitochondrial matrix. So the electrons from cytochrome C are then passed through complex 4 and finally to the oxygen and reduces oxygen to water. Now the other important thing to know is there are many electron acceptors. NAD and FAD are the electron acceptors, but oxygen is the final electron acceptor in the electron transport chain. And also it is important to remember that oxygen is the most electronegative element and it has a high affinity for electron. So because of that high affinity for the electrons, oxygen basically pulls electron through electron transport chain towards itself. So that's actually the driving force here, which is the electronegativity of the oxygen atom. And electrons have a tendency to travel from atom which are less electronegative to the atom which are more electronegative. So when the electrons are traveling from complex 1 to complex 3 to complex 4, that means complex 1 is the least electronegative, whereas oxygen is the highly electronegative molecule. And you can also think as oxygen as a magnet which is actually trying to pull the electrons from complex 1 to complex 3 to complex 4. So that's how it is actually forces electrons towards itself through electron transport chain. Now lastly, I just want to quickly review the process of electron transport chain. So the electrons which are carried by NADH, it is then transferred through complex 1 to coenzyme Q to complex 3 to cytochrome C to complex 4 and finally to oxygen. So now you can see that as the electron moved from these complexes and finally to oxygen, you can imagine oxygen as a magnet which is pulling the electrons towards itself. And once the electrons are with oxygen, oxygen is then reduces and form water into the mitochondrial matrix. And so as the electrons from complex 2 is then traveling through coenzyme Q to complex 3 to cytochrome C to complex 4 to finally to oxygen. Now when the electrons are traveling through different complexes, as I mentioned that the passing of this electron through complex 1 to complex 3 to complex 4, they actually liberate the energy and causes the conformational change in complex 1, complex 3 and complex 4, which then acts as a proton pump to transport protons across the inner membrane from the mitochondrial matrix to intermembrane space. So at one point, there's going to be more protons in the intermembrane space compared to the mitochondrial matrix, and this pumping out protons from mitochondrial matrix to intermembrane space is eventually creates a proton gradient. So basically proton gradient is when occurs when the more electrons are pumped out from the mitochondrial matrix into the intermembrane space. And this proton gradient eventually creates this pH gradient and an electrical gradient. So the electrical gradient creates more positive charge on the outside of the membrane or into the intermembrane space, whereas the mitochondrial matrix becomes more negative because there are very less protons left in the mitochondrial matrix compared to the intermembrane space. And the pH gradient lowers the pH of the intermembrane space, that is, it is making the intermembrane space more acidic, whereas the mitochondrial matrix has high pH and makes it more alkaline. So basically proton gradient creates this high energy and uneven distribution of protons across the inner membrane of the mitochondria. And furthermore, both these proton gradient and the electrical gradient tend to attract these protons back into the mitochondrial matrix from the intermembrane space of the mitochondria. So electrons are then traveling back into the mitochondrial matrix through a special enzyme which is called ATP synthase, which is also called as complex 5. So basically the flow of this proton down the electrochemical gradient into the mitochondrial matrix drives the synthesis of ATP with the help of ATP synthase enzyme. And this phenomena of synthesizing ATP through proton gradient is called Mitchell's chemiosmotic hypothesis. So how does the proton gradient synthesizes ATP through ATP synthase enzyme or complex 5 enzyme? That is the topic of my next video, which I'm not going to cover in this lesson. So thank you so much for watching, guys, and I hope you really learn something from this video of electron transport chain, and I promise that I will see you soon with my next video of electron transport chain and oxidative phosphorylation, where we will learn how the ATP is synthesized through ATP synthase or the complex 5 enzyme. Thank you so much for watching, and please do not forget to like, share and subscribe the channel, and I will see you soon next time.